Today, we're going to unravel one of the greatest stories ever told. It's the story of how we discovered, understood, and finally learned how to rewrite the code of life itself.
I mean, really think about that. It sounds like something straight out of a movie, right? But this question, it's not science fiction anymore. It is the central question of modern genetics, and it has the power to change, well, everything. So how on earth did we get here?
Well, our journey doesn't start in some futuristic high-tech lab. Nope. It starts in a quiet monastery garden with a man who is about to figure out life's most basic rules using nothing more than some pea plants and a whole lot of patience. So meet Gregor Mendel. Today we call him the father of genetics, but back in the 1800s, he was just a monk with a big question. Why do we look like our parents? He was sure there had to be a pattern, some kind of logic to it all. And to figure it out, he turned to the humble garden pea.
He started with a really simple experiment. He took a pea plant that he knew always grew tall and crossed it with one that always grew short. Now, your first guess might be that the babies would be somewhere in the middle, right? Kind of medium-sized? Well, that's not what happened. Not at all. Every single one of the new plants was tall. The short trait had just vanished. Or at least it seemed like it had.
And this, right here, was Mendel's first huge aha moment. He realized that some traits must be, well, stronger than others. He called them dominant. The others, the ones that get hidden, he called recessive. So the tall trait was dominant and it was just hiding the short trait. But, and this is the key, he had a hunch that the hidden trait wasn't gone forever. So what did he do next? He took those tall plants from the first experiment and let them cross-pollinate with each other. And guess what happened in the next generation? The short trait was back. And it wasn't random. For every one short plant, there were 3 tall ones. It was a perfect, predictable 3-to-1 ratio. This was massive. It proved that traits are passed down in these neat little packages. Mendel called them factors. Today, we call them genes. Okay, so Mendel had figured out the rules of the game, but he had no idea what was actually carrying those rules from parent to child. To find that out, we have to jump ahead almost 100 years, from the garden to the lab, to find the actual molecule that holds life's blueprint.
And here it is, the star of the show, deoxyribonucleic acid. You can just call it DNA. This beautiful, twisting molecule is the thing that carries all that genetic information Mendel was talking about. It is literally the instruction manual for building you, me, and every other living thing on this planet.
So how does it work? Well, the easiest way to think about it is like a twisted ladder. The 2 long sides of the ladder are made of sugar and phosphate, and the rungs are made of just 4 chemicals or bases, A, T, G, and C. But here's the magic trick. A always pairs with T, and G always pairs with C. That simple rule, that's the code. The entire complexity of life is written with just 4 letters.
Now, figuring out this structure was one of the biggest races in science history. And the deciding clue came from this kind of blurry-looking X-ray picture. It's called Photo 51, and it was taken by a brilliant chemist named Rosalind Franklin. Her work was the critical piece of evidence that proved DNA was a double helix, a key that 2 other scientists were just about to use.
And those 2 scientists were James Watson and Francis Crick. Using Franklin's data, they were able to build the first correct model of DNA. It was such a monumental discovery that Crick famously walked into a pub and announced to everyone, "We have discovered the secret of life." And you know what? It wasn't an exaggeration. Understanding the structure meant we could finally understand how life stores and copies its own instructions.
So, we've got the rules, and now we've got the blueprint. What's next? Well, obviously, we're going to learn how to edit it. This is where the story shifts from just watching and understanding to actually getting in there and changing things. Welcome to the world of genetic engineering.
The very first tool you need for an editing job is a pair of scissors, right? Well, scientists found them. They're enzymes called restriction endonucleases. But these aren't just any scissors. Think of them as molecular scissors that are programmed to cut DNA only at very specific letter sequences. For the first time, we had precision. Of course, if you have scissors to cut things out, you're also going to need some glue to paste things back in. And we found that too. It's another enzyme called ligase. It's basically genetic glue. And with scissors and glue, the era of cut-and-paste genetics had officially begun. And the real-world impact was, it was world-changing. Suddenly, we could do things like this. Take the human gene for insulin, cut it out, and paste it into the DNA of simple bacteria. And just like that, the bacteria start following the new instructions, pumping out human insulin.
we had turned tiny microbes into life-saving medicine factories, making treatments for diabetes, growth disorders, you name it. And it didn't stop with medicine. This new ability to read and cut DNA led to another total game changer. In 1984, a scientist named Alec Jeffreys had a brilliant realization. Just like our fingerprints, our DNA sequence is unique, and with that, he invented DNA fingerprinting.
The idea is pretty simple. By cutting up a person's DNA, you can create a unique pattern of bands, almost like a genetic barcode that belongs only to them. This completely transformed criminal justice. Investigators could now identify a suspect from a single drop of blood or a strand of hair. But it also helped in happier ways, like proving who a child's parents are and reuniting long-lost family members.
From cutting and pasting, we now jump to the modern era, which is all about searching and replacing. This brings us right up to the day where these new technologies are giving us almost unbelievable power and raising some really profound questions.
The big name you hear about today is CRISPR. If the old tech was like cut and paste in a document, think of CRISPR as the find and replace function. It's a biological tool that can scan through billions of letters of genetic code, find one single typo, and fix it. The level of precision is just incredible, and it opens the door to maybe, just maybe, fixing the root cause of genetic diseases like sickle cell anemia. But a power like this is a classic double-edged sword. The promise is breathtaking. We're talking about potentially curing diseases like Huntington's, or designing crops that can survive a drought and feed more people. But the peril, the risk, is just as big. People worry about unintended side effects, about ethics, and even about the potential for this tech to be misused in terrifying ways.
Now, all of this modern magic stands on the shoulders of one of the most monumental scientific projects ever, the Human Genome Project. This was a massive global effort, and in 2003, they did it. They published the first complete map of the entire human genetic sequence, all 24,000 or so of our genes.
And what we learned from reading our own instruction manual was, well, it was humbling. For starters, you and any other person on Earth, you're 99.8% genetically identical. We also learned we have about 200 genes that are exactly the same as genes found in bacteria. And maybe weirdest of all, most of our DNA doesn't seem to do anything. It's been nicknamed junk DNA, and it's still one of genetics' biggest mysteries.
So this brings us full circle, from a monk counting peas in a garden to having the complete map of what makes us human, and now holding the tools to actually edit that map. We literally have the power to steer our own evolution. And that leaves us with the biggest question of all. It's not a question of technology anymore. The question is no longer, can we do this, but should we?